With our current scientific knowledge and technology advancements, many have speculated about the development of gene editing and its possibilities. However, there is still a lot to learn about gene editing and the ethical grounds with it. We will explore the history, risks, prospects, and many other topics of gene editing and its ethical concerns. Learning objectives: history of gene modification, modern technology of gene modification, ethical concerns of gene modification. So when did gene editing start and how did it come to be? According to Dana Carroll in our Yale journal titled Genome Editing, Past, Present, and Future, the first targeted genomic changes were produced in yeast and in mice in the 1970s and 1980s as the first genome editing softwares were produced in the late 1900s. The first gene editing was done by Herbert Boyer and Stanley Cohen in 1973 with the usage of transgenesis. It is a mode of experimentation involving insertion of a foreign gene into the genome of an organism, followed by germline transmission of the gene and analysis of the resulting phenotype in the progeny. This accomplishment allowed for further experimentation of gene modification in 1974, where the first genetically modified animal, a mouse, was created by Rudolf Janege. This promoted the commercialization of the modification technology that resulted in a bacteria that produces somatostatin and insulin in 1978. The first genetically engineered plant was achieved in 1983 when an antibiotic-resistant gene was inserted in tobacco. A brief history video of gene editing is followed.
Humans have been engineering genes for centuries. The vegetables in a modern supermarket and the staple grains that sustain us bear little resemblance to the ancient plants they derive from. Our domestic animals, too, have been transformed through generations of breeding. The variety of dogs is a testament to our power, long before modern biology, to transform genes. Beginning in the 1970s, we learned to directly manipulate the genetic code stored in the cells of every living thing. But for decades, the technology remained cumbersome, expensive, and imprecise. Then came CRISPR. In 2012, Emmanuelle Charpentier and Jennifer Doudna discovered they could hijack a mechanism that bacteria use to protect themselves from viruses. With a piece of RNA acting as a guide and a protein called Cas9 acting as the scissors, scientists can now target, remove, and replace any stretch of the double-stranded DNA molecule in any living thing. Easy, quick, inexpensive, and so precise, scientists can switch out a single base pair in the more than 3 billion base pairs that make up the human genome.
CRISPR-Cas9 was adapted from a naturally occurring genome editing system in bacteria. The bacteria capture snippets of DNA from invading viruses and use them to create DNA segments known as CRISPR arrays. The CRISPR arrays allow the bacteria to remember the viruses. If the viruses attack again, the bacteria produce RNA segments from the CRISPR arrays to target the virus's DNA. The bacteria then use Cas9 or a similar enzyme to cut the DNA apart, which disables the virus. So how is gene editing and its ethical concerns relevant to today's society? With its developing nature and its endless possibilities, gene modification is relevant because of its capability to change the many ways we function in society, such as the taste of plants, the traits of our future generations, how we tackle deadly conditions, and many other things to better our society. However, the idea of bettering society comes with ethical concerns. With the developing technologies of gene editing, many have growing concerns about the ethics of it. According to sciencemag.org, a Chinese researcher named He Jiankyu announced he and two other collaborators forged ethical review documents and misled the doctors into unknowingly implanting gene-edited embryos into two women. Hei Jiankyu and the two other collaborators were found guilty in conducting illegal practices and were sentenced to 3 years in prison. But what are the main ethical concerns surrounding gene editing? According to the NIH in Human Genome Research, the main ethical concerns are safety, informed consent, justice and equity, and genome editing research involving embryos. Safety is a concern due to the possibility of off-target effects or edits in the wrong place and mosaicism, or when some cells carry the edit but others do not. Safety is of primary concern. Informed consent is a gray area because some people worry that it is impossible to obtain informed consent for germline therapy because the patients affected by the edits are the embryo and future generations. With our economic standpoint, justice and equity are a concern because of the probable chance that genome editing will only be accessible to the wealthy and will increase existing disparities in access to healthcare and other interventions. Lastly, gene editing research involving embryos raise concerns to many people who have moral and religious objections to the use of human embryos for research. In addition, many believe that this technology may go awry and presumably progress eugenics, or the belief in the possibility of improving the qualities of the human species or a human population by restricting undesirable traits to reproduce. With the current societal issues we have, diversity in race, culture, and background is highly encouraged. However, with gene editing, this may further endorse the eradication of diversity, especially traits of the minorities. Summary: gene editing was first experimented in 1973 by Herbert Boyer and Stanley Cohen using transgenesis. From then on, gene editing was further developed and has since been used to modify plants, bacteria, animals, and now humans. CRISPR-Cas9 has been the key to the growth of gene editing technologies. However, there are many ethical concerns surrounding this topic and how to proceed with the technology. Assess your knowledge. Which of these statements is false about CRISPR-Cas9? A. CRISPR-Cas9 was adapted from a naturally occurring genome editing system in bacteria. B. CRISPR-Cas9 is limited to simple mutations. C. The CRISPR arrays in CRISPR-Cas9 allow the bacteria to remember the viruses. Gene editing is continually being developed and improved. It is only a matter of time before we are able to reproduce newborn babies as desired. However, many ethical concerns will have to be tackled and confronted for an equal and safe chance for everyone.